All right, next we're going to go to Caleb Emmons, one of the software developers for Proteome Software. And as promised, he's going to give a sneak peek and a live demo of Scaffold Quant, our new upcoming program for quantitative proteomics, which is going to be super cool. So I will pass that along to him right now. All right, take it away, Caleb.
Okay, thank you, Jacob and Susan, and I'm glad everybody could make it to our virtual brunch, although it's too bad we can't meet in person. So as they mentioned, I'm going to be giving a demo here of Scaffold Quant, and something we've been working really hard on, and hopefully will soon be coming out so you guys can all enjoy it. So a little outline, I'm going to be talking about what the vision is for it, what types of data we'll support, how you organize your data, how you can use it to analyze protein level changes, both by looking at fold change and by using a statistical test, how you can use the program to visualize your data and its normalization, and then some more advanced options. So let's jump right into it here. Okay, so here's the program, and if it is built on the Scaffold Perspectives platform, so if you're familiar with that, some of this will look familiar. So what I've got open here is the iTrack demo from Scaffold itself, from Scaffold Q+, and I've loaded it as an MZID. So if you can export MZIDs, you can load those into this program. You can also do a direct export from Scaffold Q+ and get your data in that way. So since I've loaded it as an MZID, right now you can see it's in label-free mode, and that just means it's going to act a lot like perspectives. So you'll have spectral counting, you've got different kinds of spectrum counts here. You've got precursor intensity, you've got some non-quantitative display types like percentage of total spectra and percent coverage. And then you've got tick. This is a new feature, so we've put total ion current in for a nice analysis that way, as long as you've got spectra. And then let's go ahead and go up here and choose to enable labeled quant. So right now we've got the options to add iTrack or TMT data in here, but of course we're going to be expanding that and adding SILAC, support for SILAC, and other maybe user-defined isobaric labeling techniques. So this is an iTrack 4-plex, and you can see here there's a place to put in a purity correction or not, and let's just go with the default, so we'll hit OK. And you'll notice that the one column that we had here has split into the 4 columns of the iTrack 4-plex. So we've got the 114 through 117 columns. And now the display types available are all related to iTrack. And you can see there's iTrack intensity, log 10 intensity, log 2 fold change, or coefficient of variation. And if we want to go back, we can switch back to label free. This is kind of like switching between Scaffold and Scaffold Q+. You can just do it right there, and it's just all integrated into the same platform here. So we can switch back and forth, and that's great. So this is a pretty simple file. So let me go ahead and open up a slightly more complex one. So this is a TMT 10-plex experiment here. And let me, while that loads, pull up here. So this is from this data set. It's from a paper by Plubel, Wilmarth et al., and it's from 2017. And the data is freely available from the Proteome Exchange, PXD, and here's the identifier for that if you want to take a look at it yourself. And so this experiment was a 2-way analysis where there were 20 mice split into groups of 5 and then respectively fed either a regular chow diet or a high-fat diet. And then they were sacrificed for analysis after either 8 or 18 weeks. And the experiment has 3 MS samples, 3 runs through the mass spec, and each of those has 10 TMT channels, so we get 30 what we'll call quant samples. and it's loaded here. So you can see here the 30 samples. Now that's kind of a bit overwhelming, hard to analyze. So the first thing we want to do when we get an experiment like this open is give it some organization. So to do that, switch to the organize view here. And in the interest of time, I've gone ahead and already created the organization for this experiment. So over here, you'll see the 30 quant samples. And then here you'll see the different categories that I've created. I've created a diet category with the regular chow diet and the high fat diet. And I've created a time category with the 8 weeks or the 18 weeks. And then I've assigned those all those attributes to the samples over here. There's various ways to do that. You can drag and drop into folders. You can right click and assign things that way. So it's a pretty flexible interface here to get your data organized the way you want it, assign all the attributes. And then once you've done that, oh, I should mention actually, so you'll see there's, in these two folders, there's 10 mice. So each of this is one mouse, each of these samples, and then each of these samples is one mouse. And then there were, so that only accounts for 20 of the quant samples. And then there were some more used for pooled, excuse me, yeah, for pooled samples. So they mix together all of the other samples and include it, and that's for normalization. Okay, so the next thing is you want to tell the program how you want to view the data. So to do that, you can click here, configure experimental design, and it'll bring up this dialog. And you can see, so we can just walk through this. If you have fractionation, you would indicate that here. And if you have technical replicates, you would check this box. We don't have that in this experiment. So the next thing is to choose a design. So we can have either the basic design, a repeated measures design, or a two-way design. You may think, since there's a time component here, that this is repeated measures, but remember that's when you've got the same subjects that are measured multiple times under different conditions. And in this case, it was actually different groups of mice that were sacrificed at 8 and 18 weeks. So it's not a repeated measures, it's a two-way design. And then we can specify what's the primary analysis category. So we're most interested in diet, so we'll put that here. And our secondary category is time. So we'll put that in there. And then if we hit apply, and switch back to the samples view, lo and behold, you'll see that the 30 columns that we've seen have now gone down to 4. So we've got the chow diet broken out by 8 weeks or 18 weeks, and we've got the high fat diet also broken out by 8 weeks or 18 weeks. And then you can use this control up here to switch. If you want to say switch back to seeing all the samples, we can choose TMT sample here, and we're getting all the columns back. Actually, we're only getting the 20 that are organized in this organization, because remember that the 10 samples weren't part of the actual experiment. They were just normalization samples. So here you'll see the 20 mice samples, and if we switch, we can also roll all the way up to diet, and then we just get 2 columns for regular chow and high fat. So if we switch now back to the time one, so we're seeing all 4 columns here. Now we're ready to start looking at protein level changes. So the first way you might want to think about doing that is looking at log2 fold change. So if I select that here, in this case, now the columns are showing the fold change between, so it's the log2 fold change between whatever column you're in and what you've designated as the reference. And here the reference is set to regular chow diet. You could set the reference to more specific things, any specific sample here or any time, diet and time combination, but let's keep it on regular chow diet for now. So this will compare the fold change between, what's in this column and all of the regular chow samples. Now we might want to do a more targeted comparison between just what's in the 18 weeks under high fat and the 8 weeks under high fat and the 8 weeks under regular chow. And so this is another new feature here, which we can do if we bring up this quant dialog and look at the reference samples box here. You can choose use match reference samples. So if I select that and hit apply. Now you'll see what it's done is now it's comparing just the 8 weeks high fat samples to the 8 weeks regular chow samples, and the 18 weeks high fat samples to the regular chow 18-week samples. So that's why suddenly these are all 0, because they're being compared to themselves, and these are just being compared to the matching one. So that's a pretty nice feature. Good. So the next way you might want to compare your protein level changes is actually by applying a statistical test and seeing if there's some significance. So let's go ahead and click on this quantitative analysis button again. And the rest of it here is all about statistical tests. So we can choose a two-way ANOVA for this data, and we can apply Benjamini-Hochberg multiple test correction. And remember, with the two-way ANOVA, there's actually 3 different p-values you can get. So they're listed here. So you can look at the primary factor effect. Does diet have an effect on group mean? You can look at the secondary factor effect. Does time have an effect on group mean? Or you can look at an interaction effect. Is there an interaction between diet and time? So let's start with the primary factor effect and hit apply. And when you do that, you'll get another column here in the samples view. And in it will be p-values. And these are color-coded. So you've got white means not significant. This orange color means significant at 5%, but not with multiple test correction. And if I sort on these, you'll see sadly in this case that we don't have any that are significant after the multiple test correction. They would be green if they were. But since we've got 1,300 proteins, you end up getting quite a significant multiple test correction. Okay, so, and here right here, you also got the selector for what kind of p-value you want to look at. So right now it's under primary factor effect, but if I switch here to interaction effect, it'll switch the p-values and they will now be showing the interaction p-values. So if I again sort on them, that'll be giving me the proteins with the most significant interaction. And let's say we want to select one of those and drill into it, get some more information about that specific protein. We can then select it and click on the proteins view and say switch to the statistics tab here. And that will bring up this statistics tab, which shows a table with all of the two-way ANOVA results, all the sums of squares, and all the p-values over here. And then over here, you'll see the interaction chart, which is pretty helpful to see that, yes, indeed, there really, for this protein, was a strong interaction between diet and time. You can see that for the 8 weeks, it went up between the regular chow and high fat, and the 18 weeks one went down between the regular chow and high fat. And while we're here, we can switch to the spectrum tab, and we can see that indeed, yep, here are the TMT peaks for this spectrum. So you can get a nice look at your spectra that way. Okay, the next thing that I want to do is look at some of the visualization tools that we have available. So that's under the visualize view here. Oh, actually, I want to make sure that I'm on log 10 intensity here. Okay, so if we switch to visualize, you'll see that we've got a nice volcano plot, and we've got a quantitative scatter plot over here, so you can compare different samples that way. And then there's a graph for some protein trends, and we've got, here's a new chart that shows pre and post normalization. So this is the quant samples values before normalization, and they've been brought nicely into alignment here. There's actually multiple steps to normalization, of course, and this is sort of the first one that this graph is showing. And just to talk a little bit about some of the other steps, one of the main other steps of normalization that is currently being used is called IRS normalization, internal reference scaling normalization. And that was a technique developed by Wellmarth and others and actually introduced with this data set. So that's kind of nice. And one way we can visualize normalization is if we switch to the principal component analysis tab here, and if we look at the scores plot, so right now the data is not normalized. So if we look at the scores plot, so you'll see actually 3 really strong clusters happening here, and you might guess what those clusters are. You might guess that they're the MS samples. So let's verify that. One way we could verify that is by editing our summarization here. Bring this dialog back up. And if instead of organizing on diet and time, we switch back to a basic design and put the MS sample or biosample in as the category. If we wait just a second, this should update. And you can see here, it's color-coded very nicely that indeed the 3 clusters that you're seeing are coming from the MS sample. Now, that's a bad thing, right? You don't want to have all of your data points clustering based on what MS sample that you happen to put the quant sample in. So, and that's happening because currently the data is unnormalized. So if we switch back to the samples view here and check this little checkbox to turn normalization on, and then switch back to the visualize view and look at the PCA again, you'll see nicely that the normalization has succeeded in removing the dependence on the MS sample, and we've got just some nice overlapping circles. That's what you want in this case to show that the MS samples don't lead to the clustering. Okay, to wrap up here, I wanted to just point out a couple more advanced options that all of this is in progress, so that we're still coding it up, enabling everything, making it all work right. So here's some things we've got coming down the pipeline. For normalization, maybe you've got some internal standard proteins, in which case you want to come to this page and click add protein, and you type in the name of them, and you can select your favorite protein and put it in there. Whatever if you have 5 or 10 or whatever internal standard proteins, you would put them all in there. There's also a box here for designating pooled reference samples. So remember, this experiment did have pooled reference samples, so you would come here and you would choose the attribute, in this case treatment pool, that tells the program which ones are your pooled reference samples, so it can use those. And then we've got another tab here. Again, this is still in progress, being added to, but these are some options to choose which spectra are used for quantitation. Perhaps you want to, by default, we actually would exclude shared peptides, but maybe you want to use them for quantitation, then you can check this checkbox. Got something called reporter ion tick ratio, which looks at the ratio between your reporter ion current and your total ion current, and you can use that to exclude spectra. So, and there'll probably be some more options coming here. So I think those are the main things I wanted to show you today. And yeah, if you have any questions, suggestions, I'd love to hear them. You can contact me directly, caleb@proteomesoftware.com. So thanks for your attention.
All right, thank you, Caleb. Looks like the scaffold quant is coming along nicely.